right, good morning, everybody. Doopy-doo. All right, so let me just get rid of this and this. So what I said we'd do at the beginning of this class, although it's going to be a little truncated, unfortunately, is quickly go over that last 3-point cross, just to make sure that you're all on board with what's going on. We're going to do this kind of quickly, largely because we've got a bunch of other stuff we have to do, and the exam is next Thursday. So I want to make sure that you're properly prepared for that. Ooh, yum. Okay, so quite quickly, has anybody managed to do this, just out of curiosity? See if some of you have the answers already. Awesome. Well, that's good to hear. Okay, so move that over this way. Okay, what's the first thing to do? In your plan for success? All right, find the parent genotypes. Yeah, excessive. So anything that's the largest number, essentially, if linkage is occurring, are going to be the parentals. So those are going to be the two at the top, right? And it's going to be easy for you because essentially they are all in cis, right? So we have all wild type, all mutant are the parents or the parental chromosomes. All right, which are the double recombinants? So I'm just going to, smallest number, yeah, that's good. Which ones are those? Just going to type this stuff up. So spineless, yeah, and scarlet ebony. Thank you, everybody. Parental chromosomes are +++, STESS. Okay, so which allele is moving? Which of those is moving in that double crossover? So if spineless and scarlet ebony are the double recombinants, oh bloody hell, I didn't even finish time to say that. All right, yes. Excellent. So what's the gene order? See how fast you can type now, Tanya. Ha, not fast enough, it appears. All right, yes, exactly. So SS has to be in the middle. So it's going to be essentially +++, and then ST, SS, and E. Or, and you don't know this, right? It could be the other way around. Yeah, that's what I want. It could be E, SS, ST. Doesn't really matter for what we're doing. Either is right. Typically, we just do it from left to right as we're given. Okay, so recombination frequency between ST and SS. So what are we looking for? Which are the single recombinants between ST and SS? Yeah, we've got to have the mirror image, right? The reciprocal, but which one are we looking at? So we've got to find essentially one which is plus and then SSE. And the reciprocal of that would be ST++. Okay, so which of those? What numbers are we looking at? Okay, excellent, Gabriel. Yes. So those are these two here, right? Because we have a plus, or actually it's even easier to look at this way. Plus SSE, right? That's the first one. And the reciprocal is going to be ST plus plus. Okay, so that's RF equals, what's that? 50 plus 52 plus what? Yeah, exactly. And that's all divided by 755 times 100. So what does that give us? Okay, 14.6 centimorgan. Okay, now for the other one, which is going to be between RF between SS and E. So that's the middle one and the one on the furthest right. We need, so that's going to be plus E, and what would that be? ST, SS, and plus. It's really obvious which ones those are because those are the ones you haven't used yet, right? So you don't even really have to even work that out. You can just use a kind of a shortcut and go at it, right? So these are the ones at the bottom, right, which we haven't added up yet. So the combination frequency is this, those two, plus 3, plus 2, divided by 755 times, oops, 100 equals what? Dang, you fast. There you go. And that's all there is to it. So even kind of chuntering around and waiting for Tanya to type really slowly, it didn't take that long, right? So like the laborious way of doing it, kind of writing everything out like this, it took, what's that, like 5 minutes at most. All right, and you can do that faster yourselves. You just have to practice, right? So that this stuff is just like so, and remember the rules, and you can't get it wrong. Anybody have any questions about that before we move on? No? Okay, so we're going to take a pretty huge step change now. So we've really been looking at like the bigger picture stuff of inheritance, kind of looking at phenotypes, looking at large scale interactions between genes and alleles. What we're going to do now is we're going to shift really for probably most of the semester, to be honest, trying to think my way through everything that we're going to cover. And we're going to go much more to the small scale, right? So essentially what you've done so far is you've got a big overview of what's happening at the individual level, right? Or between generations. And now we're going to dive into what's happening at the cellular and even nuclear level, right? So really from now on, it's going to be less math, which may be a good thing for some of you. And it's going to be a lot more enzyme names, processes, kind of molecular biology, right? And really that's essentially what's going to underlie a lot of what you've been learning so far, right? So we've done the bigger picture stuff. Now we're going to kind of narrow down into the molecular biology of genetics. And then when you take evolution, which may be either next semester or the one after that, then you're going to come back out again and you're going to look at bigger picture processes such as natural selection and evolution and gene flow and things like that, right? Which are kind of, again, more math-based, right? Because you're dealing with larger numbers. So let me move this over here. And so which you prefer kind of really just depends on your own personal, pardon me, proclivities and your own strengths and weaknesses. Quite often people are either kind of big picture mathematical type geneticists or small picture molecules and proteins kind of geneticists. And so which you find easier kind of just tends to depend on, come on, which of those you fall into. Okay, so let me just get comfy. All right, so this is a picture of a essentially a replication fork, part of a replication bubble. And this would be in, oh, this may be, oh, this is actually in humans or eukaryotes. And the way I can tell is because of the names. Eukaryote DNA polymerases have funky names, and they also call this topoisomerase instead of DNA gyrase. Right, so this is essentially where we're going to kind of end up at, I guess, in probably by middle of next lecture, right? And we're going to finish. So this lecture is going to be a lot of the basics of DNA replication. So structure of DNA, types of or modes of DNA replication, because they vary from one organism to another or type of organism. And hopefully we'll get through DNA replication in prokaryotes. Right, that's the aim for today. And as I said in the email I sent around earlier, if I don't finish that, if we don't finish that today, I'll do a short kind of Zoom meeting with myself and go through the rest of what we need to cover today, right? So that you're not going to be scrambling to catch up. And then next class on Tuesday, we're going to do eukaryotic DNA replication and structure, because there's some funky stuff with eukaryotes that doesn't occur with prokaryotes. And then we're going to dive into homologous recombination, right? The actual mechanism of that, right? So we've talked about the outcome of homologous recombination a couple of times in meiosis and in linkage. And then on Tuesday, we're actually going to go into the molecular mechanism of homologous recombination, which is pretty funky, I have to say. Every time I look at it, I'm like, ooh, God, that makes my head hurt. All right, so, and then after that, next Thursday is the exam, right? So it's kind of chop, 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 chop. Hopefully next section 3 that we'll be doing after next week, we'll kind of go back into a bit more of a normal cadence, but it's still going to be pretty intense. Okay, so just a wild kind of run through basics of DNA, right? Again, this is stuff that we have covered. It shouldn't be too much of a surprise what you're seeing here. One thing I do like about this picture, though, is it shows the atoms that are used to form hydrogen bonds between complementary bases. And the reason why I like this, let me just get the chat back up. Make sure that nothing's popping up in there. The reason why I really like this is these red letters here, right? So these are, in addition to the size, right? So you can see that purines have 2 rings, pyrimidines have 1. These are the nitrogenous bases, right? Which are going to hang off of the sugar. So not only is size an issue, right? So you can't have 2 purines together or 2 pyrimidines because the DNA strand will bulge out or kind of collapse inwards, right? It has to be exactly space. So there's always going to be 2 and 1 rings or 2 and 1, whichever way around. But the reason why you don't have a guanine bonding with a thymine is because of these, right? So these are electronegative or electropositive areas on the base. And what I mean by that is kind of going back to GenBio 1 type stuff, is that this oxygen and guanine, for example, has a strong pull on the electrons it's sharing with the carbon. And so this is ever so slightly negative, electronegative. And vice versa on here, the nitrogens are kind of doing the same, right? They're pulling the electrons ever so slightly away from these hydrogen atoms that they're sharing those electrons with. It's not creating an ion, it's just creating a slight difference in charge. And if you look, these three match perfectly with, dang it, I've got my screen covering it. Here we go. These three here, right? So this is going to be slightly positive, slightly positive, slightly negative. And now we have slightly negative, slightly negative, slightly positive. Right, so we have an exact match between these two, or these three, sorry, and these three. And between these, you will find hydrogen bonds forming. This is a large part of the basis of the specificity of DNA replication. Because if you have a thymine try and bond with a guanine, right, in error, you can see we're going to have a plus and a minus and a plus. Well, that that might kind of bind here, sort of. It's just not going to fit very well, and it's not going to bind very securely, right? And so when it comes to seeing if that base is correct, it's going to stick out, right? It's not going to sit in there properly. And it's going to be, A, it's not going to go in the first place. And B, if it does, it's going to get recognized as being wrong and get cut out. Okay, so just these basic properties, right? And these are really, well, I mean, they're not basic, because this isn't an ion. See, chemist joke. Simple property is what allows DNA replication to be astonishingly accurate, right? In the order of maybe 1 to 3 mistakes every time our cells divide. And we have about 6 billion DNA bases or letters in our diploid genome, right? Not all of the accuracy is down to this. There's an awful lot down to DNA repair, but a big chunk of it is. Come on, Ella. Okay, the other thing that is super, super, super important to reiterate, even though I've kind of talked about it already, it'll go, there we go, is you can only add new nucleotides to the 3' end of DNA, because that's where that hydroxyl group is attached. And this is used to form, where'd that picture go? Oh, it's actually in this picture, nuts. Okay, sorry, I wasn't looking in the right place. Allows these phosphodiester bonds to form, right? So essentially, this upper line here, right, going between the O on the second nucleotide to this P, that phosphodiester bond, that is what is catalyzed by DNA polymerase, right? So before this base was added, say this is the newest addition to this growing strand, right? There would have been an OH just there, right? And DNA polymerase uses the energy, and there's actually a couple of extra phosphates here before it's incorporated, uses that energy to create that phosphodiester bond. And so the next one will get added just down here. That hydrogen will disappear and form water with another OH, and you'll have another phosphodiester bond here and a newly incorporated base. Essentially, that's what DNA polymerase does. All right. Any questions so far? Kind of whizzing through this a little quickly. Again, this is stuff that you kind of already know. I'm just making sure you kind of get the context. Again, also just to reiterate, 3 hydrogen bonds between guanine and cytosine, 2 between adenine and thymine. And also, okay, so another interesting property of DNA, which is going to be relevant to what we look at later, is it actually has, oh, we got 2 puppies that we've just started looking after, and they're kind of crying out. Anyway, maybe, can you bring them over to show the class? Okay, you can have a little puppy vision. Have I showed you the puppies yet? I thought it was the other class. Did I show you them to you? I did? Oh, okay. Don't worry. They're just going to get a free hug instead. Can remember who I showed them to. Anyway, one of the important things that's actually recognized by DNA binding proteins, of which there are many, is this structure, right? So DNA is not an even twist, like twisting 2 wires together or a twist tie on a loaf of bread. It is not an even twist. It is a alternating twist where you have a tightly wound part called the minor groove and a more loosely kind of stretched out twist called the major groove. And there are many proteins such as transcription factors. We'll be talking about some of them in a little bit. Initiator proteins that start DNA replication. They recognize not just the sequence of DNA, right, the individual bases, which I always think is kind of funky, to be honest, because how do they see them? They're on the inside. But they also recognize the shape. And so I can't remember. I think it's TRPR repressor in bacteria actually has a kind of this sort of shape, and each fingertip fits into a major groove next to each other, like a key, like so. And so this is also important in a lot of the stuff that we're going to be talking about. So many of these processes, right? So generally speaking, in an awful lot of molecular biology and molecular genetics, you have 2 general themes. One, we have a series of events. It's like a story. It's a beginning, there's a middle, and there's an end, right? And that applies for pretty much everything that we're going to be talking about, right? So you have to start the process, you have to do it, whatever it is that you're going to be doing, and you have to stop, right? Because at some point you're going to stop copying the DNA because you're going to run out of DNA to copy, right? Also, the second main theme is that these processes are carried out by complexes of proteins. So it is not just one protein doing something. It's not like the catalysis of a reaction, right? Turning, I don't know, glucose into fructose, for example, right? Carried out by one protein, one enzyme. We have multiple actors. Sometimes they act sequentially. Other times they act like in like this big agglomeration of proteins together to perform a function. Individually, they don't work. Together as a group, it's actually called a holoenzyme, which we'll come to later. If I can get my chat box back up. So that's an enzyme that is made up of many proteins. And DNA replication is kind of like that. Transcription is very much like that, which we're not going to get to for a little bit. But just to get this idea of, we're not just talking about single proteins doing their stuff anymore. We're talking about lots of proteins acting in concert. either sequentially in time or in cooperation as a group at the same time. So those are themes that you will come across many times over the rest of the semester. Okay, and so let's just, I'm gonna move this down actually a little bit, if I can. Yeah, I don't know. I know that's kind of out of place. I'm not worried about that for now. Okay, so some notation or terminology. We have what's called an origin of replication. This is in bacteria. Hey, come on. For example, this is just a slide we'll get to later on or on Tuesday. This is a region which is recognized by proteins which begin the replication process, right? The whole kind of initiation of DNA replication, right? So that's one kind of point to get. In bacteria, there's only ever one because they have a circular genome and you just have to start at one point and do it. In eukaryotes like us, there can be thousands of these per chromosome because we do not replicate our chromosomes, start at one end and go down to the other. It would simply take too long, right? We do it kind of multiple places all at the same time. So that's one thing. And the other is that you will end up with a replication fork. Actually, there's replication fork and there's replication bubble, right? Essentially, a replication fork is the leading point at which the DNA is being unwound, and that's kind of moving forward down the original DNA double helix, right? And you will have, for example, here, a whole bunch of proteins at that fork doing stuff. DNA polymerase, helicase, et cetera, right? Now, once you expand those, typically, not always, but typically those forks kind of expand away from each other, right? So the DNA gets opened at the origin replication. Now in the kind of corner, you have your replication forks, and those will expand until you have a replication bubble. That's basically is just, you know, those two replication sites kind of moving further apart as DNA replication proceeds. Okay, so these are kind of words that we're going to be using a whole bunch. Another point that's also really important to get across is that essentially when you, so this, again, this would be a replication fork just here, right? We're not showing the enzymes, but we're showing the original parental double helix gets separated to one parental strand here and one down here. And each one of these parental strands serves as a template for the synthesis of a new daughter strand. And this process continues. Essentially, it's going to go up the page on this slide until all of that original parental double helix is separated and is now paired with a newly synthesized daughter strand, and the other one also with a newly synthesized daughter strand, right? And so that's called semi-conservative replication. It means that in each new chromosome, right? This is one, or chromatid, right? Before DNA replication, DNA division. And this is another chromatid. This one is going to consist of a parental and a daughter, right? So half of it is new, half of it is conserved. This one is also going to be half new, half conserved. That's why it's semi-conservative. You don't have too old and too new, or the other way around. because you're always going to be using one of the original strands as a template to synthesize a new strand that matches it. And that's what you can see down here. Obviously, once this cell goes through meiosis or mitosis or whatever, this is now going to be the parental, and this will then get split into two, and each one of those gets copied for the next round of cell division. Okay, any questions so far? This is also a really neat picture because it illustrates the major problem in DNA replication. This is an anti-parallel double helix. See, you got a 5' end up here, and that goes down here to 3' end, right? 3' end goes down all the way to here to the 5' end. Now you know that DNA can only be made in the 3' direction for the new strand. So we have a replication fork going up the page, like so. And we have one strand, this one, is being made in the same direction as the replication fork is moving. Because you can see here, this is the 3' end. So we can add, next one will be a C, or next one will be a G, a T, A, whatever, right, that matches this template, this parent, going in the same direction. This one is not, because the parent strand is going 5 to 3' up the page. So the newly synthesized strand has to go in the other direction down the page. So this strand, this daughter, is being synthesized in the opposite direction to the replication fork movement. But both of these daughter strands have to be synthesized at exactly the same rate. Otherwise, you have all kinds of problems, right? You have presence of single-stranded DNA in your genome, which is just bad news, right? So we have to fix it so that this is called the leading strand because it kind of goes with the direction of replication fork movement. This is going to be called the lagging strand. It's not lagging because it is synthesized more slowly. That's a really important point to make. It's lagging because it takes longer to finish. And you'll see why later on in a bit. Okay, so a lot of the contortions and kind of issues in DNA replication revolve around this conundrum, right? The fact that these two daughter strands are being synthesized in opposite directions, and yet they all have to move together in the same direction with the replication fork. Okay, you'll see how we fix that in a bit. Okay, so quickly going to go through modes of DNA replication, right? Because they differ quite markedly, actually. And once we get through that, then we'll be able to just quickly kind of go over this whole leading, lagging strand and the like, right? So we should get that done easy in the next 15 minutes. And then probably I'll actually use my kind of like solo lecture to go through a chunk of prokaryotic DNA replication, right? Because we don't actually have a whole bunch of class time left before the exam next Thursday, right? So I want to make sure that we cover everything properly. And also, obviously, I'm around for if you have any questions that you want to send by email or even meeting up individually is also fine. Okay, so bunch of alphabet soup. We're going to have a whole bunch of enzymes required. These each do different things. They are very specialized in what they do. Even the DNA polymerases are very specialized in what bits of DNA they synthesize. We obviously need single-stranded DNA as a template, right? So we need to be able to pair up new nucleotides with an existing strand, using that as literally as the template to stick them to. And we need a whole ton of DNTPs, right? So these are our incoming nucleotides that are going to get incorporated into the daughter strands. These last 2 points are one of the main reasons why you have the gap phase in gap 1, in cell division, because you have to make all of these, right, before you can actually synthesize DNA. So that takes time and energy. Okay, so essentially we have different ways of synthesizing DNA or replicating DNA based on the different types of DNA or genomes that different organisms have, or different things. So the simplest and fastest is what's called rolling circle replication, right? So some DNA viruses, right? Not all of them, because some of them are linear, but some DNA viruses are circular. And also we have these things called plasmids, right? And so plasmids are basically little circular chromosomes that bacteria can share with each other. And they typically contain stuff that is not essential for bacterial life, but stuff which can be really advantageous. So you'll typically find things like antibiotic resistance genes or virulence genes, or, kind of other funky stuff on these plasmids, right? So that's one way in which antibiotic resistance can spread through bacteria. It's not just you're selecting for a novel mutation when you give someone some antibiotics or give animals antibiotics. You're also selecting for the spread of these plasmids, which confer resistance to bacteria that previously weren't resistant. So they're pretty funky things. Anyway, these are replicated by what's called rolling circle replication. And essentially the idea behind this is that you peel off one of those strands while you're synthesizing the new one. So you cut, or nick is the kind of term, one of those two strands, and then as you use that, say that inner strand as a template, right? That's what that red one is, the new strand being synthesized. You kind of push off. It's like you unwrap the strand that it's replacing, right? And that strand then gets, was originally linear, right? Because it was cut, gets resealed, and so now it's circular, and that then serves as the template for the synthesis of the other strand. So you get this, right? This was with the original intact circular strand, right? So that gets you your completed plasmid or viral genome. But you also get these extras, right, which you kind of peel off, right, in the process, and those also get replicated, so you have more copies, right? And the benefit to this is speed, right? So this mode of DNA replication does not suffer from the anti-parallel problem that we talked about earlier with the leading and the lagging strands. Every strand is leading. Every strand that's being synthesized in rolling circle replication is leading. And also you can continue or repeat the replication of one strand before the other one is finished. So you can finish this one and you can punt off this other strand to be replicated later while you're finishing up this one, right? And then once you finish that one, that might be working, and then you just repeat the whole process for the one that you've just done. So it's very, very fast, which if you're a virus is a big deal, right? Because you want to replicate your genome and package it into proteins and stuff before your host kills you or before your cell dies, right? You've got a limited amount of time to be able to replicate and kind of get out, essentially. And so that's really where rolling circle replication succeeds. However, this is really just for plasmids of essentially small bits of DNA, because it gets kind of complicated as stuff gets bigger, right? So this is for maybe stuff that's up to 10,000 bases in total, right? And kind of the size of the piece of DNA. Now, bacteria, so their circular genomes are maybe 200,000, 500,000, you know, they're in the hundreds of thousands of bases, right? So they're much bigger, right? Still nothing like a eukaryote. They replicate using what's called theta replication. And so the reason why it's called that is because that looks like the Greek letter theta, right? And some wag who is, you know, looking at this with an electron microscope is like, oh, look at that. I learned Latin at school, and called it theta replication, or Greek. I don't know. I did learn Latin at school. Can't remember one diddly little bit of it, to be honest. It's the most boring subject I've had to study, apart from math. Anyway, so in bacteria, we have a single replication origin, or origin of replication. It's called an ORI-C. And then you can have either uni or bidirectional replication. And so essentially the replication fork and the machinery are the same in both. But in unidirectional, you just have one replication fork, right? So it's going to go all the way around kind of counterclockwise until it finishes back at the beginning, and you get your two bacterial genomes. Or bidirectional, where it goes in both at the same time. I don't actually know why you would prefer one over the other, to be honest. I don't actually understand that. Maybe it's kind of the amount of resources that are present dictates one over the other, or how fast you want to replicate, right? And bacteria in the right conditions, like E. coli, can replicate once every 20 minutes. So it just goes to show how fast this can be done and how relatively little DNA they're going to be copying. Some other bacteria, which is crazy, some bacteria actually replicate around once every 100 years, which is kind of crazy. Like some real extremophiles that live in very challenging environments, you know, to us and to them, presumably, replicate so slowly that they appear like they're dead. And they're not. They're just growing very slowly, which I think is pretty cool. So in DNA, in eukaryotes, it's very different, right? And the reason is eukaryotic chromosomes are, one, they're linear, right? So they're not circular. They're just one long piece of DNA. That actually causes other issues, obviously, later on. But also they're huge. And we're talking in the millions of base pairs each chromosome, right? Our whole genome, our haploid genome as a human, is about 3 billion base pairs split between 23 chromosomes. And so you cannot have one replication origin per chromosome. It would simply take too long. Even as it is, it takes, you know, mouse cells a day, 24 hours to divide, right? And a big chunk of that time is spent doing DNA replication. And so in eukaryotes, you can have thousands of these replication origins per chromosome. And so you'll essentially have thousands of bubbles, right, which are moving kind of essentially, you start with bubbles here, 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 right? And those slowly get bigger and bigger and bigger, which is what you see down here, until they hit one. And then they simply merge, and then you get a larger replication bubble. And that keeps going and going and going until all of the replication bubbles have merged, and now you have your two sister chromatids. So that's the way around the issue of having such huge bits of DNA to copy, is they copy them in lots of places at the same time until they're all finished. Like kind of dividing the labor up, I guess. Okay, anybody got questions? No? Okay, good. It's relatively straightforward so far. Right, so in talking about leading versus lagging strands, right? So in bacteria, you're going to have that issue, right? If whether it's uni or bidirectional replication, right? So each replication fork is going to have a leading strand, right? And the leading strand is essentially synthesized continuously, right? So without break from that origin of replication. And then the lagging strand, what it's actually done, and there's some cool videos out there that you can find which will illustrate this for you, is that, thanks, love, is that this template is folded back on itself. And so essentially these arrows, they're actually synthesized in the same physical direction, right? This is how that problem is solved. But you can only do that for a little bit until you hit what you've already done. So this would be the first one. And then you flip it and you synthesize it. Essentially, you're walking backwards, right, into the future. Then you start the process again and you synthesize a little bit. Again, that arrow is actually pointing in the same direction as the leading strand as it's being synthesized. But you hit what you've already done. So you stop. And then you fold it back again and you do another little bit. So essentially you'll be synthesizing this little bit here. Again, you can only go so far walking backwards until you hit where you've come from, and you'll hit this one and so on, right? And so these, and this is the same in eukaryotes as well, these little chunks, right, which vary in size from tiny in eukaryotes, like in the hundreds of base pairs, to small in prokaryotes of 1,000 or 2 base pairs, right? Compare this to a chromosome which can be 100 million base pairs long. You can get the idea of why this is called the lagging strand, right? It takes a long time to stick these things together. But this lagging strand is going to be synthesized in small chunks called Okazaki fragments after the scientists that discovered them. My daughter just found a flea on one of the puppies. Anyway, a little bit of drama aside. And so they're going to be potentially in eukaryotes, thousands of these, right? Remember, these two strands are synthesized at exactly the same rate. But if you look at the leading strand, there's very little stuff that you have to do to this, right? All you have to do is stick it to this, fill this gap in here, right? The lagging strand is made up of a large number of chunks, and each one of those chunks has to get stuck together to the one next to it. Otherwise, your chromosome, when you go to replicate it again, will consist of a lot of small chunks, which just isn't going to work. right? That's why this is called the lagging strand, because you have to go back after DNA replication has proceeded. That replication fork has already kind of moved further off the page, essentially. Once that has moved on, you have to stick all these individual small fragments together so that this is one continuous strand, just like the leading strand is. And that takes time. Okay, so that pretty much finishes this one. I'm going to post this and a second small video later, probably around lunchtime, give or take, or maybe this afternoon, depends on when I can get to it, where I'll go through prokaryotic DNA replication, right? So make sure you look at that one, and any questions about that, bring that with you to class on Tuesday and we'll go over those. Unfortunately, I have to run. So now you can have a lovely weekend, right? Now this time I got it right. And I'll see you next week.